So, hi everyone, and welcome to the fourth TIA Centre seminar of the academic year. So I'm delighted to introduce Dr. Spencer Watson today. Spencer started his research into the tumor microenvironment in the lab of Professor Joe Gray at Oregon Health and Science University, developing novel high-content bioengineered assays to study microenvironment, mediated resistance to targeted therapeutics. Following his PhD, Dr. Watson relocated to the University of Lausanne in Switzerland in the lab of Professor Joanna Joyce. There he studied immunotherapeutic approaches to targeting the immune microenvironment of gliomas. During this time, he also worked as part of the Cancer Grand Challenges IMAX team to advance techniques in spatial biology, high-dimensional imaging, and digital pathology analysis. The title of Spencer's talk today is Investigating Glioblastoma Recurrence with Spatial Multi-Omics. So yeah, Spencer, thanks again for joining us, and we're really excited to hear your talk.
Yeah, absolutely. So I'll get started. So yeah, thank you for having me here today, and for everyone for either tuning in or being there in person. So yeah, as you said, the topic of this talk is using spatial multi-omics and tissue analytics to investigate, especially the role that fibrosis plays in promoting glioblastoma recurrence. Oop, let's see, will it let me advance here? There we go. So a quick bit of background on glioblastoma. I'm sure everyone's very aware this is the most common and devastating tumor type that arises from within the central nervous system. And the typical standard of care treatment for glioblastoma, especially high-grade glioblastoma, is surgical resection followed by radiotherapy and temozolomide-based chemotherapy. But despite these treatments, the median survival time for patients is still less than 14 months from the time of initial diagnosis. And what makes this such a really challenging disease to treat is this huge amount of plasticity and heterogeneity that exists within the tumor cell population. And that's really going to complicate any treatment that's trying to just specifically target just those cells alone. So this is why a lot of the field is moving towards also targeting the more genomically stable tumor microenvironment, in particular the immune component of the microenvironment. So we have lots of trials ongoing now to target T cells and dendritic cells. So in the lab of Professor Joanna Joyce at University of Lausanne, one of our main targets was tumor-associated macrophages, especially trying to treat them with what are known as anti-CSF1R inhibitors, such as this agent from Novartis, BLZ-945. And the goal is to re-educate the TAMs from a pro-tumoral phenotype back into an anti-tumoral phenotype. And in preclinical mouse models, this was really successful. So we could shrink down even very large tumors down to small dormant lesions. And so the growth curve here at the top, you can see that we get really rapid regression in the first 7 to 14 days, after which they enter a period of what we call dormancy, centered around, as we define, 28 days after treatment. But what we saw was about 44% of cases, these tumors would rebound under treatment. But what was really interesting about all of these cases is that 100% of rebounds, every time the tumor recurred, it always regrew next to a glial scar that had formed during treatment. So this led us to the hypothesis that these glial scars weren't just some byproduct of the treatment, that they were actually promoting tumor recurrence. And so it followed, if we could inhibit the formation of these scars, we could potentially improve the response to the therapy. So just a tiny bit of background on glial scarring, because this is different than like scarring in the skin or other epithelial tissues. So following mechanical damage or oxidative stress or really any severe insult to the CNS tissue, you get waves of cellular responses that result in a kind of multi-compartmental type of scar. So along the border of the scar, you get what's known as the glial part of the scar. And that's where you have these hypertrophic reactive astrocytes. They increase in size and number, and they link up together to form a cellular barrier against the site of damage. But if the damage is severe enough, at the core of this, you'll get this dense accumulation of fibrosis. And so this sort of spatial compartmentalization of scarring in the CNS becomes really important when we try to assess its role in the tumor microenvironment. So our model for studying glioblastoma is what's called the RCAS model. And this is a viral transfection model where we drive overexpression of PDGFB. But because it's a viral model, we can also include other modifications. And in this case, we added GFP so we could track where the tumor cells go. And looking at some of the MRI volumetric data on the left here, we can see that following the initiation of treatment, when we get scarring, all that's left at the side of the tumor is this large, dark, contiguous area. And kind of importantly, when we have recurrence, it's always next to these structures that we see regrowth. And then using correlative IF, we were able to determine that these structures in the MRI signal were made up of really densely packed ECM proteins. So in particular, like collagens, fibronectin, and tenascin C. And these were bordered, again, by these sort of wall of hypertrophic reactive astrocytes. But really importantly, we didn't only see this scarring response to just this anti-CSF1R treatment. We also saw it following more standard models of treatment like radiotherapy or surgical tumor resection. And we also saw it in other models. So not just this RCAS model, but we also saw it in this HRAS GFAP-CRE model and in GL261. And also kind of importantly, these tumors form in a different anatomic region of the brain. So it seems independent of where in the brain as well. So it kind of paints the picture of any treatment that is aggressive enough to really debulk the tumor has the potential to trigger this wound healing glial scarring response. So to try to validate this in the patient setting, we teamed up with some neurosurgeons at CHUV University Hospital in Lausanne. And so the way this worked is when a patient came in for surgery for their recurrent glioblastoma, we had the opportunity to sit down with their MRI data and then identify regions of putative fibrotic scarring that was related to the treatment of their initial primary glioblastoma. So then we could go into the operating theater with the surgeons and identify these specific areas for analysis. And by doing this, we were able to see a very similar association between tumor regrowth and this fibrotic scarring that was related to the initial treatment. So we wanted to try to assess what the cellular protein composition of these scars was and how they form. So we turned to a few different omic profiling technologies. And to try to get some of the temporal formation of these scars, we assessed multiple time points following treatment. So the key at the bottom of these figures shows which time points were assessed by which modality. So we started with single-cell RNA-seq, looking at samples from untreated, comparing that to 7 days and 14 days post-treatment. We also did a really large-scale mass spec proteomic study where we did manual microdissection to try to isolate individual regions from each of the tissue sections, and then pooled these together for mass spec proteomics. And this made for a few different useful analyses. So if we were doing GSEA analysis, we could then cluster and aggregate these different enriched gene sets and then do a comparison not just between different time points, but between different regions in the same time point. But despite this sort of limited spatial resolution, these are still disaggregated approaches. So they give you a lot of depth, but it doesn't give you that single cell level spatial precision that you need to really assess something as complex as the tumor microenvironment. So we turned to a couple different spatial omic approaches. And for one, we did spatial proteomics using a variant of cyclic immunofluorescence that we developed for this project, and that's called hyperplexed immunofluorescent imaging, or HiFi. And in addition to HiFi, we developed a digital pathology workflow that included machine learning annotation of all the different structural features in the tumor. So regions of fibrosis or vasculature, necrosis, the leading edge, so that when we assess the individual single cells in the image, joys of giving talks from home. When we assess the individual single cells, we could look at where they are in relation to each other, but also where they are in relation to some of these larger macro architectural features in the tumor. So to give some spatial resolution to gene transcription, we turned to the Xenium in situ platform from 10x Genomics. And we initially started with their mouse brain gene panel, and then supplemented that with 100 extra genes to look at all of the different pathways and cell types in the tumor microenvironment they were interested in. So I guess anyone who's done any of these approaches on their own know that on their own, they're all very powerful and they're all very useful. But for us, the real strength of these came from integrating them together. And our strategy for that was kind of twofold. We had what we call pre-integration, where the results of one experiment would inform the design of another. And then after that, we had the more, I guess, what you call classical now post-integration, where we're actually combining the data, the single cell data from all the different modalities to create a multimodal, multi-omic data set. So I'll go through just a few of our sort of integration strategies for these data types. So between proteomics and high dimensional imaging, the proteomics was essential for identifying some of the antibodies that we want to include in the antibody panel for the high dimensional imaging. And this also helped us to validate a lot of the spatial proximity or the spatial localization, especially the ECM and fibrotic proteins that we saw in the proteomic data. And between the single cell RNA-seq data and the spatial proteomics, so the single cell was great for telling us what were the cell types that we should be assaying in high dimensional imaging. So that also informed the antibody panel. But this also worked going in the other direction, where we could take the cell-cell nearest neighbor distances from the digital pathology and then use that to inform cell communication analysis on the disaggregated single cell data. And I'll go into that a bit more later. Another thing that was really critical for us was combining the spatial proteomics and transcriptomics. And we did this a couple different ways, and one of which was physically. So following the Xenium run, we would take those slides and then perform hi-fi staining on those, along with the whole digital pathology workflow, so that we could have both protein and transcriptomics on the same image. And then we could also, because we have all these different machine learning annotated structural regions, we could see how gene transcription related to proximity to these different structural regions. So that was very useful for us. And then finally, I think probably the most useful thing in terms of data integration was combining the single cell RNA-seq data with the spatial transcriptomics. So the single cell data was great for telling us, especially what genes should be in this add-on panel that we use for Xenium. But it also meant that to a certain degree, we could project whole exome transcriptomics onto comparable cell types in the spatial transcriptomic data. And in the spatial data, because we had proximity to these different structural regions like vasculature, fibrosis, we could create spatial signatures and then project that onto the disaggregated single cell data. So that was very useful for a number of things. Next, I want to go into what we basically were able to discover by analyzing this set. But if you guys wanted to ask any questions, let me know if you have any at this point. Okay.
I don't think there's any questions at this stage. Thank you.
Okay, great. I can't see anyone. I closed that window, so you have to pipe up if you do. Okay. So first, we wanted to look at where were the surviving tumor cells after treatment. So looking at the 20-day dormant time point, the only place we were able to find surviving tumor cells was specifically in the fibrotic part of the glial scar, in particular in regions that were high in tenascent C. And so using the Xenium data, we were able to determine that these cells within this spatial niche were very highly expressing of a range of different dormancy and stem-like genes. So I guess the clinical implication of this is, cells that are expressing dormancy and stem-related gene signatures are more likely to be resistant to any treatment that's going to target neoplastic cells. So radiotherapy or chemotherapy would likely not be effective on cells in this environment. And to look at some of the immune component of the environment, first looking at macrophages. So in this model, we have a huge amount of macrophages in the untreated context. But following treatment, these same TAMs, again, these are the cells that we're actually targeting with the anti-CSF1R inhibitor. We can see that these are basically excluded from regions where the surviving tumor cells are. And looking at proximity data from HiFi, we can see that this sort of like disassociation of these two cell types increases more and more over time until by the point of dormancy, they're almost mutually exclusive. We actually kind of have the opposite story with T cells. So In the untreated context, we have very few T cells in the tumor, but following treatment, we get this huge influx of T cells kind of specifically into this fibrotic niche. But looking at what's called Project TILS analysis on the single cell data, so looking at T cells after 7 days of treatment or 14 days of treatment, we can see that all these T cells in the fibrotic part of the environment all have exhausted signatures. So they're not effective in actually attacking the tumor cells that they're near. So taken together, this kind of paints the picture of the fibrotic part of the niche being a pro-survival spatial niche. It's holding tumor cells in a dormant state, and it's protecting them from immune surveillance by macrophages and T cells until conditions are optimal and they're able to regrow out in the brain and then cause tumor recurrence. But this idea that these dormant tumor cells in the fibrotic niche are the cellular source of recurrence is something we wanted to validate. So we looked at the xenium data for samples that had rebounded under treatment. And what we did is take all the tumor cells and we binned them into 4 different concentric regions emanating away from the region of fibrosis that formed during treatment. And looking at especially the first 2 concentric rings, we can see that these had very similar gene expression signatures when compared to tumor cells at the 28-day dormant time point. So they had a lot of these same dormancy genes shared between them. And this similarity got less and less as we move away from the fibrotic niche. So it seems as if essentially once cells escape out of the scar, they maintain a lot of their, I guess, gene transcription similarity. But as they regain proliferative capacity, they change and evolve as they sort of move out and invade the brain. But this does really seem to support the idea that this dormant cell population in the fibrosis is the source of recurrence. But actually, we don't really know how the cells are escaping the scars. So we're still working on that. So whether it's maybe the ECM is being degraded by the environment, or I guess through stochastic migration, these cells happen to find their way out into the brain. Kind of however it happens, when the cells do escape the fibrotic scar, they find themselves in a very growth promoting environment. So using some of the regional proteomic data for the scar versus adjacent rebound tumor, or scar versus adjacent healthy brain, we can see that the fibrotic scar is highly expressing a range of different mitogenic paracrine growth factors that can essentially, once cells are in this environment, they've regained proliferative capacity, they're just awash in wound healing growth factors that can cause really dramatic growth. And this is actually what we see when we go back to the volumetric data, that at the point of tumor recurrence, we get really aggressive regrowth that can sometimes be much, much faster than when the tumor formed in the first place, which shows that so the fibrotic part of the glial scar is not just promoting tumor survival, it's also potentiating and promoting more aggressive tumor regrowth at the point of recurrence. So now that we've sort of narrowed in on the fibrotic part of the scar as being this survival niche, we wanted to see what cell types were key in driving the formation of this structure. So we focused in on this population of cells that were transcriptionally similar to vascular pericytes, that for the sake of hedging our bets, we're just calling perivascular-derived fibroblast-like cells. And especially this cluster of PDFLs on the right had very high expression of collagen 1 and fibronectin and all these ECM-related genes. And they were also uniquely associated with the post-treatment samples. So we didn't see this phenotype in the pre-treatment samples. And so using the proteomic data to create a gene signature for specifically fibrosis, we could see that this PDFL cluster had the highest unique expression of this gene signature. So we next want to see what were the signals that were driving this PDFL cell type to respond and create all of these ECM proteins after treatment. So we use a cell communication tool called NicheNet. And I guess if anyone's done any cell communication analysis on disaggregated single cell data, you know that you get a ton of different, very significant cell communication events. So it becomes really hard to tell what are actually the most important ones. So what we did was leverage some of our cell-cell distance data from HiFi and created a probability matrix of two cell types being within 30 to 50 microns of each other in the actual tumor environment, and then use these probabilities to weigh and filter all of this long list of cell communication events to try to find those that were like just the most biologically relevant. So focusing on the PDFL cells, that's the blue receiver cells on the top of the chart, we can see that a range of different cell types are expressing different neuroinflammation genes like IL-1B and also TGF-beta 1 and 2. And this is driving the specific expression of some of these ECM proteins that we saw in the fibrosis gene signature. And to validate this, we went to our combined spatial transcriptomic and proteomic data set. And so here we're using ERTR7 as a marker for PDFL cells. This is typically a marker of fibroblast-like cells in the CNS. And then using the xenium data to find cells that were the most highly co-expressing of the TGF-beta receptor and the downstream collagen 1 gene, we can see that there's a very high association between these two cell types. So this really sort of picked out this PDFL population as the driver of this fibrosis. And these cells were interesting for a number of reasons, and one of them was their time dynamics. So in the untreated context, we really don't see many of these cells at all. Like there's maybe a few associated with like large vessels, but almost immediately after treatment, just like 1 or 3 days after treatment, there's this like explosive growth of this cell within the lesion until about you get to like 5 or 7 days, and they basically spread out and encapsulated the entire region in this fibrotic ECM. So some people may know that there's an idea that's called a type A pericytes. It's been around for a few years, and it's a cellular population that typically exists along the vasculature. And then in reaction to any sort of damage to the CNS, they'll detach, they'll delaminate from the vessels. They'll undergo waves of proliferation and differentiation to stage this fibrotic wound healing response to any damage to the brain. So this seems to be very much what we're seeing here in the post-treatment glioblastoma microenvironment. It's just amped up to 11. So looking at the single cell data and looking at some GSEA analysis, we're able to see some genes that kind of fit this idea of type A pericytes. So we have big upregulation at 7 days of EMT genes, extracellular matrix organization, interferon gamma response. But what was interesting is that when we then looked at a later time point, when we compared 14 days post-treatment to 7 days, we could see that now a lot of these same gene signatures are now down-regulated in this comparison. So we took these gene signatures and made these gene pathways and made some gene signatures for EMT, ECM, interferon gamma, interferon alpha, and then track their expression over time. And what we saw was this huge increase in all of these gene signatures between untreated and 7 days. But after that, they all either plateaued or spontaneously abrogated on their own. So there was I think there's quite a lot that's interesting here, but for us, the most important takeaway is the clinical implication of this. So what this is telling us is that if we want to inhibit this PDFL cell's response to treatment, we have a very narrow window of intervention, just between the first 7 to 10 days, that we're actually able to achieve any sort of inhibition, because after which these pathways will fall off on their own. So it's very unlikely that any inhibition post this point is going to be effective. So we developed a treatment strategy using a combination strategy to target both neuroinflammation and TGF-beta in combination with our anti-CSF1R inhibitor. And we actually had tried many, many single agents throughout the course of the study, and none of them were able to successfully really impact glial scarring after treatment. So for neuroinflammation, we went with a very broad inhibitor. So we used dexamethasone for the first 2 weeks and then tapered off. And for TGF-beta, we used a very specific inhibitor. We used galenistrib for just the first 10 days. So this worked for a couple key reasons. And first, it's targeting this narrow window of intervention for these PDFL cells. But this also gets around a lot of the kind of extreme toxicity issues that you get with using these agents long term. So looking to do some validation work at just 14 days post-treatment. So there's some example images at the top, and the bottom left is a quantification of the amount of fibrotic area after treatment. So we can see adding just dexamethasone to BLZ doesn't really do anything in terms of blocking fibrosis. When you add galenistirib to BLZ, we can see it impacts it a bit, but it's not significant. There's still plenty left after treatment. But adding all three agents together resulted in an almost complete elimination of fibrosis after treatment. And the chart next to that, that's the quantification of the amount of tumor cells remaining after treatment. And you can see there's this really high concordance to the amount of surviving tumor cells and the amount of fibrosis in the lesion after treatment. So this, based on this, we did a long-term progression-free survival study where we tested all of these combinations of agents. So here with the red line, you can see this is BLZ 945 on its own. And similar to before, we're getting about 55% survival. When we add dexamethasone and galenistirib on their own, so no BLZ in this case, you can see that this does nothing in terms of survival. Unfortunately, they die very fast as if we were only giving them vehicle. So when we add dexamethasone to BLZ, again, we're not getting much of an improvement. The same with galenistirib. But when we add all three agents together, we're able to very significantly increase survival from 55% to 95% survival. In fact, we only lost one mouse in this arm of the trial. And so this kind of gets back to the core of our hypothesis that this fibrotic glial scarring response to treatment isn't just some benign byproduct. We can now see that the fibrotic scarring part is a pro-survival spatial niche that's holding cells in a state of dormancy. It's protecting them from immune surveillance, and it's then, at the point of recurrence, potentiating and boosting their regrowth. And now by inhibiting the formation of this response, we were able to very successfully improve the efficacy of this particular immunotherapy. So moving forward, we're really interested in now translating this to other sort of more common methods of treatment like radiotherapy and surgical resection, but also potentially other immunotherapies as well. And then ideally, eventually move this into the clinic. So just as a summary here. So what we were able to do in this study was integrate several kind of large-scale multi-omic approaches, including single-cell spatial transcriptomics, single-cell disaggregated transcriptomics, mass spec proteomics, spatial proteomics, and then combine them all together to basically investigate this fibrotic response to treatment. And again, what we thought was really important was that this wasn't unique to this anti-CSF1R treatment. We also saw it following radiotherapy, and following surgery. We saw it in other models, and we also found evidence for it in the patient setting. And so by analyzing the full, complete spatial multi-omic data set, we were able to identify this fibrotic niche as promoting a dormant stem-like phenotype for surviving tumor cells. We could see that it was excluding the phagocytic macrophages that we were treating with the drug, and it was highly associated with T-cell exhaustion. And then by producing all of these wound healing growth factors, it was boosting regrowth at the point of recurrence. And I will say that it was really the full, complete multimodal data set that was super critical in achieving this sort of last aim, where we were able to identify what the cellular driver of the fibrotic region was, and then identify what were the cell signaling events that were driving that response so that we could develop an inhibition strategy to specifically target that. And by doing that, we were able to block this fibrotic response and improve the efficacy of the anti-CSF1R treatment. So with that, I'd just like to thank everyone that was involved in this project. So yes, you can imagine with so many different modalities, it was quite a large team. So we're very lucky to have very talented and dedicated researchers. Chief among them is Professor Joanna Joyce at University of Lausanne. So all of this was done in her lab, and she was absolutely critical to the success of this. I'd also like to thank Anouk Zomer. She was my partner on this project. We were co-first authors on this publication. So you can see the cancer cell cover for most of this project that I presented here today. So if you'd like to read more about it, you can check it out there. And also, I'd like to thank all of our funding agencies, and then everyone for joining the webinar today. And I think I spoke much faster than I wanted to, so I think I have plenty of time for any of questions. So open it up to any questions you might have.
Brilliant. Well, thank you very much, Spencer, for a fantastic talk. That was really, really interesting. So, should we open the floor to questions then? Does anyone have any questions they'd like to kick us off with? Or shall I start us off? Oh, have you got a question?
I can ask one then. I'm just wondering, to what extent would you say some of the processes you've identified as happening in these glioblastomas, to what extent do those processes perhaps generalize to other cancers? Do you think the similar things are happening in other, or is it very, very specific to brain cancers?
I think how it happens might be specific to brain cancer, but this fibrotic response is actually kind of ubiquitous across a lot of cancers. So my previous background was in breast cancer, and we absolutely saw this there. And then also sort of try to like appreciate more the impact this had on immune treatment. But it's just that the treatment like modality was different, because typically in breast cancer, you would do neoadjuvant treatment. I mean, in that case, almost triggering fibrosis is a benefit, because then you come in, you take that out, you clean that all out. But because of how you have to treat glioblastoma, we don't have that option. So definitely we see it in breast cancer, I think a bit more in colorectal and other sort of epithelial tissues. Yeah, I think, but the actual like molecular mechanisms, I think are quite different.
Yes. Brilliant. And we're a computational pathology group. So can you give us a bit more information about how you go from, you've got this clear treatment effect from 38 mice, right? So how do we go further forwards to take this to humans and eventually to take it to the clinic? What are the next steps, I suppose?
I think next steps immediately are translated to something that is more commonly used in standard of care. So anti-CSF1R treatments been in clinical trials, but those have so far not been successful. We have limited evidence of this in our models that we didn't publish on the effect of radiotherapy. But in that case, patients already receive dexamethasone alone to deal with a lot of the sort of fluid buildup resulting from radiation. So that's not an agent that we can actually, translate effectively. So I think, yeah, moving this to a treatment modality where we nail down, basically, I think our idea is that each sort of like environmental perturbation might require its own tailored anti-fibrosis strategy. So I think that's where we want to go next, is looking at radiotherapy, looking at surgical resection, and then from there, trying to move towards translation.
Brilliant. Sounds really exciting. Does anyone else have any questions?
I have so many questions, but I'm a little bit afraid to ask because I'm too ignorant to understand lots of the great stuff that you talked about. So first of all, thank you very much for the great talk. So as Adam mentioned, we are mostly computer scientists trying to do cancer research, so not as knowledgeable as you are. So I'm just speaking of my own ignorance about just getting my head around one of the first things that you mentioned. So you said that previously you would have thought that these scars, these are like byproducts of tumor recurrence in brain. But whereas your research, the way you presented it, shows that they actually are they have a role in tumor genesis and recurrence. They actually promote it. So I couldn't. So can you, like, in layman terms, explain again how does that happen? Because I was thinking about this, and I was thinking maybe it's not related to fibroblasts at all. Maybe there's another confounding factor, because you said that the tumor starts to regrow from the same place that the surgery happened, and I'm thinking, for example, maybe it's because the tumor was not removed properly in those cases, right? So there's like some tumor residual left there, and that ignite the whole process again. Or for example, it's something else with the whole brain structure, or the genomic of the tumor itself that forces it to start growing again in a certain area of the brain, right? So it was also.
Oh yeah.
So I'm sorry, I'm trying to fit in all my question into one. So I was also wondering if, for example, you looked into the cases that they have not formed, tumor rebound, and what was difference between these two, to see if you can identify any confounding factor there.
Right. Okay. I'll start with the first one, and you might have to remind me of the second one by the time I get through it.
Yeah.
But essentially, no, you're absolutely right that a lot of what classically we've chalked up to tumor recurrence and glioblastoma happening at the site of the initial resection is because we are not getting the full margins, and you do get disseminated tumor cells away from the tumor bulk. But because that's sort of been the dogma, surgeons and researchers haven't looked at fibrosis. And one of the challenges we faced was that surgeons threw out fibrosis. They didn't send it to the pathologist. They didn't biobank it. They considered it as waste product. And that was sort of the general consensus, is that this is just nothing. And so very little research has been done on it because of that. So we're probably most likely looking at two things. It's not just that tumor cells are escaping the resection boundaries of the surgery, but also they are surviving within some of these fibrotic niches. So we're having essentially a two-pronged issue. So, I mean, I think one of the things I always say about this is this isn't the mechanism of recurrence, but it is a mechanism of recurrence. And it's one that we've just sort of overlooked for a number of years. So now it's not just our group that's done this. We've also had some recent papers that have come out, strictly genomics, finding that ECM gene signatures were highly predictive of faster time to recurrence and worse overall survival. So this is something we're more and more convincing neurosurgeons and maybe hopefully the research community that this is actually something worth exploring, because even if you solve the problem of disseminated tumor cells, you still would have this problem. And now I knew I would forget. Please remind me the second part of your question.
Forget about my second part because I also forget it. And but no, I guess one more important thing
was what was the difference between the ones that didn't recur?
Oh yeah.
Yes.
Yeah,
we did that as part of this project as well. So every time we had a tumor recurrence, there was a glial scar, but the converse wasn't true. There sometimes could be glial scars where we didn't see tumor recurrence. So that was chalked up to a couple different things. So some of that was because this model has a constitutive tumor knockout to it, we have laws here in Switzerland that we can't take it past this 16-week time point because they'll spontaneously get lymphomas. So we're limited by the time course. So because they, the ones with scars didn't develop a recurrence by the end point, but maybe they could have afterwards. But a lot of what we saw is we didn't get this dense deposition of ECM. We had glial scarring, but we didn't have this fibrotic core. And so that seemed to be the real key difference. And there was also a lot of differences in the amount of sort of immune inflammation in the environment. The scars tend to be very cold and hypoxic. So scars that may have formed but didn't develop this sort of like chronic scarring phenotype allowed other immune components to get in and still attack the tumor, which either would have prevented recurrence or delayed it past the endpoint of the trial. But no, it's still something that's very interesting. It's challenging in the in vivo model to further explore. So we're looking at now in vitro bioengineered ways of trying to recreate these conditions so we can ask some of these questions.
And thank you very much.
Does that answer your questions?
Yeah.
Brilliant.
Of course. Sorry.
No, they're great. They're great questions. Great questions.
Does anyone else have any questions? Agree?
Just another quick question related.
Of course. Please go for it.
Yeah, please.
Mostly related to my first question. Did you have any record or measure of tumor aggressiveness before the surgery to see if there is a correlation between the ones that have rebounded or not?
We We do, but so far we haven't seen anything that really matches it. The problem is we are a little limited in getting basically cases that come through our local hospital, which is one of the larger hospitals in Switzerland, but we don't get hundreds of patients. I mean, we have maybe hundreds after 5 or 6 years of collecting. So when we start to account for all the other independent variables like age, methylation status, IDH, wild type, or mutant, our N gets very small, and we're not really able to do any statistical analysis with any real power. No, but it's very interesting, and we're trying to assess how can we predict whether or not a scar is going to form. How can we predict recurrence if there is a scar? It's just something that in the patient setting we just need more data.
Yeah, makes sense.
I suppose there's a bit of a naive technical question from me. So in some of our collaborators are quite interested in generating lesion level mass, tissue maps essentially.
Yes.
And so to get the sort of images that we've seen in your slides, it looks like it's not just one or two whole slide images that you've got the tissue from. So how have you gone about sort of stitching these images together?
Yeah, because we do whole slide imaging, and basically what we developed HiFi for was to be able to do it at high throughput, so that we could have up to 100 plus slides in a single experiment, try to cut down on like batch effect. So yeah, this is basically tiled IF scanning microscopes. And then the stitching is done either in system using the Zeiss Zen platform. And we've worked with Zeiss a lot to really customize. It's kind of like every step in the process. I think we're in talks now, like putting out an actual product based on this. So hopefully it becomes a lot easier for everyone else. You have to go through the same headaches that we did to have to optimize it. But it's essentially ensuring that we have complete flat field uniformity across multiple rounds of imaging for each single section of tissue. And then we developed I think an alignment pipeline tool that's up on GitHub now that can align at subpixel resolution all of these images.
Oh, brilliant! That sounds great.
Yeah, that was there's a paper that came out in April about hyperplexed immunofluorescent imaging that had all of the tools and the cell segmentation models and everything that we used for this larger project.
That's very cool. And so do you have any final questions for on the group? All good? Well, I think with that, Spencer, we'll bring the seminar to a close, but thanks again for joining us. It's been really interesting.
Absolutely.
And, yeah, best of luck with your future work. Hopefully we'll come across you again. So
great. Thanks so much. It was great presenting and talking to you guys. Thanks,
Spencer. And just a reminder to everyone else that our next seminar is in 2 weeks' time. It's on the 9th of December. We'll be joined by Hanwen Zhu. So this is the author of Gigapath. Okay. So thanks again, everyone. And thank you, Spencer.
Absolutely. Thanks, everyone. Have a great day.
See ya.